All right, I'm Peter Bubenik, who wants to talk about statistical topological data analysis. Take it away, Peter.
All right, love it. So, statistical data analysis, we're interested in what it means is we would like to assemble a global topological structure from discrete data. And in particular, we'd like to extract low-dimensional representations of this structure. So here's standard pipeline for TA. We start off with raw data, which is pre-processed, and then there is an optional step in which we are transforming the data. That we want to construct a filtered simplicial complex, and typically we do that through some geometric construction. Then homology, which module, and from that the topological summary, and at the end we'd like to perform some sort of statistical analysis. So for talk, I'm going to be in the bottom half of this picture. Okay, we are most interested in for this talk here. Now, I've introduced TDA through an example that I've been thinking about recently. This is joint work with Paul Bubenik and Ezra Miller at Duke, and his student Sean Square at Chapel Hill. Looking at magnetic resonance images, and a particular type of MRI called MRA, it's magnetic resonance angiography, and it's sensitive to the motion of fluids. Lying still in an MRI machine, the thing that's moving around inside your head. So that's what shows up in these images. And one of the reasons that you might want to do that, well, first of all, this arterial structure in the brain changes as we age, so we'd like to understand those changes. Hopefully, we'll be able to use the structure to detect and locate tumors, and also to predict stroke risk. So our images, we extract here using a tube tracking algorithm. And to point out, these arteries don't have constant thickness, so these tubes vary in their thickness. In addition, there's one important feature here that's been kind of removed in the pre-processing. So something called the circle of Willis base of the brain that has some analogy, but that's been removed pre-processing. What we have here is a graph. So the data is discrete. So there's these pixels that we take as being the vertices. It's on the order of 100,000 vertices. Each of them has 3 spatial coordinates and real thickness of the tube at that point. I'm going to do some statistical analysis on these structure, but that's very difficult to do because geometry is somehow too complicated, and in particular, it varies too much between individuals. So there's no nice, simple way of aligning the brains on one subject to another and then comparing various measurements. That's very hard. There's no nice, obvious way to do that. Being topologists, we could say, well, let's simplify geometry by doing topology. Now, directly, the topology is really too simple because this structure splits up into a number of contractible components. So the homology type is just a number, and here we see the number is, these are color-coded in this picture. And for most people, that number is either 4 or 5, and that doesn't tell you very much. Intermediate, and this is where persistence comes in. So at graph, we look at a sequence of graphs, and we obtain that by looking at sublevel sets of graph. So I throw away 3 of our 4 coordinates, and we just use the height to filter our graph. And then we apply homology, and the only interesting homology is in degree 0, so we get this sequence of vector spaces and linear maps. So what that look like, well, here's one of our brains. So let's start filling up the arteries with blood from the top. And we notice when we do this, so as we move up from the bottom, certain components are born, and we're on this merge, and more go up, components are born and they merge. So we have these births and deaths of components, and persistent homology keeps all of these and organizes them in a finite set of birth-death pairs that completely captures all of the persistence module. Look at that in more mathematical detail. And since the data is discrete, we really have we always have a finite set of simplicial complexes. But technically, it's often convenient to think of that as being a snapshot of a continuous filtration. So we think of having for each real value a simplicial complex, and for pairs of real values, we have inclusions. And can go from the continuous to the discrete by just sampling on a grid, evaluating on a grid. And similarly, we can go from the discrete to the continuous in this simple way of doing that for this example. Now, if you like to think of this more abstractly, the language of category theory does that for us very nicely. If we look at this simple array of objects that has a total order on it, or n plus 1 objects, we can think of our discrete filtered simplicial complex as just a functor from this index category into simplicial complexes, or we want to into the category of topological spaces, and the continuous that is just a functor from the post set of real numbers. Now, one nice thing of this language is that we see that we can go from the continuous to the discrete by taking a pullback, and also go from the discrete to the continuous by taking a left con extension. And all of one of these objects is a given level sets of a real valued function. G in some fixed degree with codes in a field, we get the formalism, except we have now vector spaces and linear maps between them. Example here is homology of sublevel sets of some real function that need to be continuous. The great thing about persistence modules is that they have some very nice algebraic structure. So in there, the imposable objects are exactly the persistence modules that are one-dimensional on some interval, with identity maps within that interval, and are 0 elsewhere. Any persistence module can be written uniquely as a direct sum of interval modules. So for you, this set of intervals completely characterizes a persistence module. A set of intervals or multi-set is usually called a barcode. So for the data, this is what the barcodes look like for a few of the subjects. We've gone through the first 5 steps in this flowchart for one kind of real example. And the thing that remains to be done is to do statistical analysis. So let's think about that. So from a statistical point of view, we can abstract away all of those first 5 steps into function. And I maybe start a little bit earlier than the raw data and think of the coming from some underlying probability space, and the barcode is a function from that underlying probability space that comes into our set of barcodes, and we can put a trick on this set and of being pushed forward the under probability measure into this space of barcodes. I've written summary instead of barcode because later we'll look at different summaries than the barcode. All right, so summary, we want to apply standard techniques from statistics and machine learning. And this area, and they have lots of techniques, and I think have lots of methods that could hopefully combined with ideas from TA. Things like to be able to do include calculating averages, testing hypotheses, calculating correlations, applying principal component analysis. We do classification and clustering. So as it turns out that the set of barcodes does have nice metrics. One in particular is the particularly nice one is the Wasserstein metric. Where the space is complete and separable, so we can ability theory, and we can do statistics. Things are easy to do. One can do hero clustering since we have a metric. We do permutation tests, which are very nice. Seem to be very hard to do. Example, calculus is very difficult. Nevertheless, there's been some nice work in this direction done by Yuri Maleko, Kate Turner, Liz Munch, Paul Bendich, Shai Mukherjee, John Mattingly, and John Hare. What we like to do is to do statistics in Euclidean space, which is easier. So we can, if we can convert our barcodes into vectors. There's a few that have been proposed. We at the lengths of the longest n bars, that gives us n numbers, standard numbers. We can look at the coordinates of the longest n bars. Functionals tau ij. These are from the coordinate ring on barcode space. These have been studied by Garrelson and his collaborators. But for talk, I'm going to look at one way of doing this, which I call the persistence landscape, and has advanced that it doesn't lose any information and that it is stable. So persistence module, there's a continuous and a discrete version of the persistence landscape. The continuous version is a real valued function on n cross r, or a sequence of functions from r to r. And we get a discrete version of that, or just a vector, by implementing this function on a grid. This function, the definition is very simple. It's this definition here. Let me give you, show you what this looks like. So if we can draw a barcode here. let me give a value of t here. So this is up here. And k is equal to 2. All right, corresponding value of the persistence landscape. So how we look at intervals centered on t, and what's the largest interval centered t? So it's a subinterval of 2 of the intervals in the code. All right, so interval there. And half the width of that interval is the value of the persistence landscape. So I calculate it from the definition for values of k and t. But this doesn't make any sense of what it looks like globally. Give a graph of this function. Let me give you a different construction. It's going to start with the program, which is of the endpoints of each of the intervals in the barcode. Now I'm going to use another function that's from the persistence rule, which is the rank function. If we take a point here, 8, so rank function corresponding to that point. So this is the rank of the map in the persistence module from filtration 4 to filtration 8. And the way that we do that is look at the upper quadrant of this point and counting the number of points in the persistence diagram that are within that quadrant. So here that the count is 1. So the count is the same for all of this region here. So let's put the number 1 in there. And for this region here, the rank function has the value 1. Here you have 2. Out here, value of 0. Value of 2. And a small triangle here, value of 3. My numbers here gives us the values of the rank function. And actually, most theory that I'm going to discuss in this next little bit also applies to this function, which is simpler than the persistence landscape. But it has some other advantages, so let me show you how that we construct that function. So the first thing, I'm going to rotate this graph 45 degrees. Picture here. And the axes here. So the vertical axis is death minus birth divided by 2. So half-life. And the horizontal axis is birth plus death divided by 2, which I call midlife. The thing we notice from this view is of all right. Yes.
It should be a birth plus death over 2, not birth plus 2 over 2.
Okay.
That's what I was, yeah.
All right. There we go. Thank you. The bases of the triangles give us the barcode corresponding to this persistence diagram. So if you want to start off with the barcode instead of the persistence diagram, you just draw these right angle triangles on top of them. And fill numbers by however many of them are overlapping. The thing that I want to do is to listen to a sequence of functions, and we do that by, let me here. So the first function we just get by tracing the boundary here. So landscape function, and the second by taking the boundary of the function, and we just continue like that. Past the maximum number of overlapping intervals in the barcode, then all of these functions are going to be 0. All right, sure, look at that. I like the 3D view of the same function, which looks nicer. So going to our brain arteries, is what our persistence landscape looked like for a few of the subjects. And I mentioned is that this is stable. So we have similar stability theorems for persistence diagrams. We have a L infinity version, which is 1, and it's almost identical to the one we have for bottleneck distance. But to point out that this actually has no assumptions on F and G whatsoever. It does not even require the Q-tame condition, which you need in order to construct a persistence diagram. I have an LP version of this stability theorem, and again, this is very similar to the theorem for Wasserstein distance of persistence diagrams. Working with functions or vectors, averaging is very easy. We just do it pointwise. And show you how this compares with averaging of persistence diagrams. I'd like to explain what the top left diagram here is. So, enough overlapping persistence diagrams that have been rotated 5 degrees. The first one of the red square, so that's one of my persistence diagrams. And the second one consists of the green triangle, so that's my second persistence diagram. To average those two, there's 2 ways I can do that. I optimal pairwise matchings, and one of them is this matching here, and then I take the midpoint of those, and that gives me the sort of the averages of those 2 persistence diagrams. Another pairwise matching that you can take, which is this one here, and you take the midpoints of those, then you get the blue ends. Here we see that there's 2 fresh of those 2 persistence diagrams. So in contrast, if we look at persistence landscapes corresponding to the red rectangles is landscape here, and for the green trials we have this persistence landscape here, and those pointwise, then we get persistence diagram here. Now, one to be a little unsettling at first is that this persistence landscape does not correspond to persistence diagram. However, this is not something that should be surprising. Typically, when we average things in a certain class, we get objects from a bigger class. So, for example, if we average integers, the average is not necessarily an integer. We average values of a true function, it's not necessarily true or false. So for example, we like the average family has 2.1 children, or that 51% of that are born are boys. Convert these back into numbers of the original class isn't really kind of loses information. So that's a useful point of view of looking at these persistence landscapes. So returning to the brain arteries, this is what the mean landscape looks like. And other that I've calculated, one about Euler characteristic of Gaussian random fields, so one about homology or persistent homology. But if we look at the average persistence landscapes of these random fields, they have this beautiful structure. And I'd love to know any theorems on this structure, but as far as I don't have any yet. So let me explain a little bit more what I have here. So this is homology in degree 0, and on the right we have homology in degree 1. So it's into higher and redo this calculation. Here we have 0 of additional Gaussian random field. If we look at used homology in degrees 0, 1, and 2, this is what the average persistence landscapes look like. So let's go back to a little bit of theory. So these persistence landscapes as living in an LP space. LP space here. So random variable, we can think of them as random variables with values in a separable Bubenik space. And that's for developing the theory. In particular, it turns out that in a separable Bubenik space, if the expected norm of your random variable is finite, then there's a special point in your Bubenik space, this E of lambda, with the mean expected value of our random variable. And it has a wonderful property, which is that if we project down to the real numbers by any linear functional, and think of that now real valued random variable, it's the exact same thing as just projecting down that special point from our Bubenik space. As our samples increases, our average landscape will convert to this landscape with probability 1. And if we look at the difference between these two functions suitably normalized, then it converges weakly to a Gaussian random variable. So for the last part of my talk, I'm going to look at 3 applications or examples. The first one is a synthetic one. So we have 1,000 points sampled instantly from the of a torus or a sphere in 3 dimensions, and they're scaled so that they have the same surface area. Now, so we have data in 3 dimensions, and most people, when they find data, they construct a filtered simplicial complex by using something like the Riplex variant thereof, like the witness complex. Now, just to do something a little different, which is closer to what statisticians do, and to first construct a kernel density estimator. Something simple, we put a little bump centered at each of our data points. Just take the sum of those functions. It's called the kernel density estimator. And then we have that function to filter a triangulation of our underlying space. This is what 3 steps of that look like for the points on the torus. So as we're moving along, we're getting more and more of a triangulation of the underlying space. And I use simplices, but I think if I had to do it again, I would use cubes. It's maybe more natural to do this with cubical complexes. All right, times for the torus and the sphere, and we calculate the expected landscapes, and this is what they look like. We have here, so on the left, in the left column is the left coming from homology in degree 0, in the middle we have homology in degree 1, and on the right we have homology in degree 2. Is the sphere, and the other row is the torus. And the challenge to you is to try to tell which one is which. So I have to look too carefully. It's maybe better not to look very carefully. So maybe stand far back from your screen or squint a little bit so that it looks blurry. And the largest features in these pictures. So large peak, here we have 1, here we have 2, here we have 0, here we have 1, and here we have 1. I give you a big hint as to which one is which now, as we've recovered the Betty numbers of the torus and the sphere. So here we have the Betty numbers for, not the sphere, the torus, and at the bottom we have the Betty numbers for the sphere. I was going to do statistics, so I really should try to verify this with some standard statistical tools rather than just by eyeballing it. Let's do that here. And I'll do a formal hypothesis test when we're going to test the hypothesis that the volume under the curves is the same. And we see that in degree 1 and 2, we cannot reject the null hypothesis, but for the middle one, the middle dimension where they really are different, where they're different Betty numbers, we do reject the null hypothesis. All right, now that data was really, so let's make it a little nastier and harder to deal with. So we throw in some Gaussian noise on both the torus and the sphere, and just noise that it's hard to tell which one is which. So it turns out right here is the sphere, the one left is the torus, and I've actually given you the top-down view of the torus, which makes it easy to see the hole in the middle. All right, and now let's redo the calculation. This time let's only use 10 samples for each, again, to make it a little bit harder. Here are the average persistence landscapes, again, with degree on the left, 1 in the middle, and 2 in the right. The same one, they're a little bit different, though somewhat similar. The 2, they're quite different, and this is because even with the Gaussian noise, there's a large void in the middle of the sphere, and that void is somehow much bigger geometrically than the void in the middle of the torus. But I think this is a good point to stress that persistent homology is not sensitive to topology. It's really sensitive to geometry, and this is a geometric difference that we're noticing here. So let's just test. This time, let's do something that's a little more sensitive than last time. Let's take the difference between the 2 landscapes and take the 2 norm of that and check to see if that's statistically different from 0. Not surprising is that we reject this null hypothesis for degrees 2 and 1, But what's almost shocking is that we also rejected for 0. That in 0, they're almost identical. So what's wrong with our test here? No, and I mean, this is something that I thought when I first ran this test, maybe I did something wrong here, because these look identical, but when we look at difference, we notice that they're actually shifted. One is slightly shifted from the other. So this test is picking up that difference. It's surprisingly sensitive. In arteries, here we have the mean shape that we had before, but a bit of variation in the data, and we'd like to understand that variation. And one way to do that is to do principal component analysis. So the first principal component is the first loading vector is in a direction that maximally explains the variation in the data. So vector looks like this. And if we remove the variation in that direction and repeat, we get the second loading vector and the third vector and the fourth vector and the fifth vector. Now, one of the things we wanted from this data was to get the correlation with age. And the earlier previous studies had quite a bit of trouble because of aligning the brains. Aligning between different subjects was quite difficult. So they had a coefficient of 0.25, which because a 1 is perfectly correlated and 0 is perfectly uncorrelated. And they felt that it really should be higher than this. So using topology, one can do better and use the landscape functions and take their one norm. We get value of around 0.5. Actually, ignore the biggest landscape function. We do better. So somehow that one is a bit noisy. And we do best by just using the fifth landscape function. And instead of using the one norm, we use the first principal component. We do even better. Like to look at is coming from protein data, and this is joint work with Gi-San Ho and her student Violetta at the University of Alberta, together with Dragan Nikolic, who's at JPL at Caltech. Important protein in E. coli, and it shuttles sugar molecules across the cell membrane. Under its functions, it has various shapes that it takes, or conformations is the word for it. And here you see two of them. We have a so-called closed conformation, and this one contains a sugar molecule inside. And right, we have a open, and it's a little hard to see why it's open, but it has a bit of a clam shape to it, and that's why it's called open. So the little bit like this here. So. So conformations of this protein have been precisely just using protein crystallography. Proteins are made up of 370 amino acid residues, and we have the exact spatial coordinates of those points for these conformations. And there are 7 closed ones and 7 open ones. Hollow means that they contain a sugar molecule, and apple means that they do not. So we have the amino residues, which are points we think of as points representing our molecule. And so the obvious thing to do is to construct a Vietoris-Ripps complex from the spatial coordinates. So that does not work very well. And something a little more sophisticated. So these proteins are actually constantly vibrating and bubbling. And instead of looking at a static description of them, instead we looked at a dynamic model constructed elastic network model, and using this model, calculated correlations between the movements of the residues. And that gives a distance in the structure, or we get a distance matrix. We project that down to 3 dimensions. This is what we look like, what it looks like, and we calculate the Vietoris-Rips complex, and what it looks like. And one thing that's noticeable here to a topologist is that we have real one homological structure here. Okay, so there's this large non-trivial cycle appearing in our complex that we'll get to in a little bit. So, calculations for all of our molecules, for all of our conformations, we can calculate average persistence landscapes. This is what they look like. And hopefully you notice some differences between these. And what we can do is look at the landscapes individually and calculate the distances between them, the norm between those functions. So I think this is the one norm between those those functions are projected down into 3 dimensions. So if we use the 2 norm, we get something similar. And left here we have this coming from homology in degree 0, and on the right we have homology in degree 1. The closed and the open conditions seem to be nicely separated using this technique. So the last thing that I'd like to point out about this data is something that's part of exploratory data analysis, and I think this is definitely a strength of TDA, that's something that it can be used for as exploratory data analysis. And now for this Vietoris-Rips complex, this is maybe an earlier stage where this large loop hasn't filled in yet. But remind you, let me fill in this cycle here. What have determined is that among these residues, there's certain ones that are more important than the others. They call them active sites, and these are most involved in protein binding. And we're aware of this, that the active sites seem to be clustered around the most persistent loop, or representative of the most persistent cycle. And those that are not clustered around that are actually clustered around the second most persistent cycle. So I think this is certainly a hypothesis that is worthy of some further study. Potentially pretty exciting result from the point of view of finding higher topological features in real data. Any of these ideas for the persistence landscape and related constructions have been implemented. The packages I'd like to make you aware of, one of which is the persistence landscape toolbox that's been developed by Pavel Dlako. And also there's a R called TDA that's been developed in parallel by Brady, Jisoo Kim, Fabrizio Lezzi, and Clement and Maria. So it was a try. And I don't go into too the details of the algorithms and the computations, but I would like to give one nice illustration here of why these ideas might be, how these ideas might be useful computationally. If your interest is not so much in the statistics, but more in other directions. If we have persistence, one of the main things we'd like to be able to do between them is calculate distances between them. And not only this is computationally can be quite expensive if we have a large number of birth depth persistence diagrams. Have here, which is on a log scale. Bottom scale here is the number of birth depth pairs. Or intervals in the barcode. And the other axis here we have time in seconds, and the computation time using state-of-the-art algorithms for calculating Wasserstein distance. I think this is using code, and Pavel's code for landscape distance. And there's a huge speed up for using landscape distance. Similar graphs for using the Wasserstein distance and the landscape distance, or the bottleneck distance and the landscape distance. I'd also like to point out that there's been some great work that's a follow-up to the work that I've presented here. The top group at Carnegie Mellon has really nice papers in which they bootstrap to calculate confidence intervals for persistence landscapes, which is really nice. And certainly they also have a really nice paper in which they convergence results for the landscapes of subsamples. And this has great potential to be a fantastic topological summary for that's going to be calculated very quickly for very large data sets. And I'll end by thanking my collaborators and my funding agency. Thank you.
Thanks, Peter. That was wonderful. Are there any questions for Peter? If you have questions, please unmute yourselves and ask them. If you, like Jen Gamble, do not have a microphone, you can type them in the chat and I'll read them out for you.
Okay. Hi. So the last data set you have with the proteins, initially that using just the coordinate of the amino acid doesn't work very well, but instead you did some magic to transform it into pairwise distance metrics. Go a little bit detail into that formation.
A little bit more detailed with there's more detail that I'll be hiding.
Okay.
So for crystallography, we know exactly where these 370 points are in kind of one frozen structure. But our analysis, we weren't able to tell anything useful from that. So saying that it's not possible to do that, just kind of, we weren't able to do that easily. So instead what we did is we used something called the elastic network model. So biophysicists have these models for protein structures that are built up using some physical principles. And they're able to kind of say how the individual pieces of the protein move under thermal fluctuations. A model, one can calculate the correlation between of any two particular. So we're calculating a 2-point correlation function, and then so that's to give us a between -1, either anti-correlated, 0 is uncorrelated, 1 is correlated. We take the magnitude of that and subtract it from 1. That gives a metric that we use. So that gives us then on this 370 points. So we have a distance matrix 300 by 370 distance matrix, and then we can calculate diagrams using like Vidit software, for example.
Basically, the filtration is still RIPS filtration, but it's on this transformed distance matrix.
That's right, yeah.
So actually, that's where I'm interested, because it's also a little bit counterintuitive in a sense that if you show in the slide showing on the left that, if we're just using the geometric embedding of the amino acids, it looks like possibly there has to be a void in between there that kind of contains a sugar molecule. But somehow you're saying that it's not easy to directly use the geometric information, but somehow the transformed distance matrix is an elastic network model. Actually, that distance matrix captures this void better than the geometric embedding.
I mean, I think this sugar molecule is much, much smaller than the protein. So there is a big void in the protein for us to see.
Oh, so this is just me kind of guessing, in a sense that it's possible that the elastic network model and the derived distance matrix can amplify this particular signal to make that void much more easily detectable than the geometric embedding.
You're welcome.
Are there questions?
If nobody have a question.
Go ahead,
Well,
Dave.
how about, yeah, go ahead.
So I have the question regarding the earlier one, the second, when you're running the PCA over a collection of landscapes. I was just maybe a little bit confused by the detail there. Are you turning each landscape into a vector and treating it as a high dimensional point and just run PCA over that?
Exactly. So,
okay.
So just sampling it on a grid, and that. Maybe a pure mathematician might be a little uncomfortable about sampling a function on a grid, but these are piecewise linear functions, and like practically the persistence diagrams that we're using were outputs, I think, from Perseus, for example, which by default from 0 to 100. So if you kind of double that 100-point grid, that determines exactly the persistence landscape. So actually the 0 to 100 here on the bottom, and then we also need to grid also needs to go in the k direction. So it ends up giving us a pretty big vector. I think we would have 250, so like a vector. That's easy to do PCA on. That's still very quick.
Oh, and a follow-up question in the same line where you give the example where you're doing hypothesis testing of the sphere or the torus. Can you say, you mentioned that, okay, when you introduce uniform noise, Gaussian noise, that all three hypotheses is rejected. But it makes me thinking that when you're actually in the very smooth case, the non-noisy case, even if I just put one single outlier in the center of the sphere, that is also going to change the landscape drastically, right?
So it depends how you construct it. So I was a little careful, and I used this kernel density estimator approach. So the one point is not going to make much of a difference in the middle of the sphere because it's going to be seen by that kernel to be very small. It's going to appear very, very late. So this is the picture here. I'm filtering by super level sets of the density estimator.
I see, okay.
So that density in the middle of the sphere, even if with one point in there, is going to be very low and it's going to appear very late.
Since the kernel density estimate is able to be a little bit robust to outliers, but it's not going to be helpful if I have uniform Gaussian noise across the data.
Sorry?
So the current density estimate is able to sort of eliminate outliers or the effect of outliers, but it can't eliminate the effect of uniform Gaussian noise. Is that?
It definitely reduces the effect of outliers, and but out of noise, it's not going to make the noise go away. It's just going to smooth it.
Yeah. All right. I'm done with my question. Thank you.
Thanks, Ben. Sure.
So in the brain, did it make a difference whether you filtered by Z or not by Z? Because that's the one you guys chose, and it makes sense of being ordered the way it is. If you went left to right, would that change things?
Very good question. So
for
this
you,
is from the beginning of the talk. So
right.
Choose Z. So I mean, it's kind of a natural choice going from the bottom to the top, but it's certainly not the only one you could make. You could go from left to right. You could go from kind of outside or outside in. So we have this four-variable function, and there's ways you could choose to filter it. So you could kind of pick any vector in free space and filter it in that direction, or even take any four vector and filter it in that direction. And
right.
We did by others, and they all work out almost the same way. So it's not important at all.
I'm sorry, just trying to remember back. I thought you said something earlier when you were looking at that. It's saying that you didn't have good alignments between the different things. Do you have a good alignment from bottom but other directions?
The alignment that I was talking about. So top, I guess where the head might be positioned there might be slight differences from one subject to another, but that's not what I'm worried about. The alignment that I'm worried about is if you want to statistical analysis on the graphs themselves.
Oh, you're meaning this bit of a brain vessel should go to this bit of brain vessel?
Right. So the earlier study
okay.
I was referring to did try to do that. I just to like exactly as you say, saying this brain part of a brain vessel in one subject corresponds to
You're meaning the global alignment.
Yeah, so aligning various branches of the tree, there's no way to do that.
Mm-hmm.
You might want to align leaves of this graph from one subject to another, and there's no way to do that.
Are there conventional landmarks for these sorts of brain vessels?
Yes. So I mean, I think at the Willis, at the base of the brain, I think there might be, the structure is a little more ordered and predictable, and that might have been the reason that was removed in pre-processing, to kind of get rid of any of those effects that might have already been understood. I don't think there are landmarks that
Actually, I was going to add a little bit here. I was talking to Steve some time ago, and he mentioned that there can be correspondences on the surface of the brain that coming from the measurements. Like, I think it's, I forgot the name, EEG or something, where there is maybe 128 correspondences surface of the brain. You might be able to infer correspondences of those brain artery trees if I say there is a point on the tree to the point on the surface of the brain.
Okay.
But it's going to be exact.
Okay, that's very good. So I wasn't aware of that. So I guess there is stuff that you can do. I think it's still, I mean, still not as nice as you might like. So certainly landmarks in the brain, and the brain structure is known, and I mean, there's various parts of the surface of the brain that do and can kind of map back and forth between them. But I guess this arterial structure is more complicated than that.
Yeah, there's not going to be any interior correspondences.
Time for one more question, if there is one.
So
The long question that's come on that.
Jen has a question, but I just also wanted to say that,
Right.
I mean, this TA approach is really cool. I mean, we're not using anything at all from the brain. We're just doing something very, very simple and kind of picking the simplest thing that we could do with it to get percent homology. And the fact that it seems to be giving better results than people who looked at it carefully using other methods, I think is a good sign for our technique. Sorry, go ahead.
Saying suggestion on chat that you can read out, or I can read out for you.
Yeah, I put it on my screen, but then it went away. So
from Jen Gamble. If put values from the kernel density function onto the triangulated manifold, don't have to know a priori which underlying manifold you're working with. How do you deal with this when comparing the torus and the sphere?
So I mean, so this is a low-dimensional thing, and it relies on knowing the underlying manifold in this case is R3, or Q sitting in R3. So there's probably cases where you might not have that, but with dimensional data, I mean, you can just we're triangulating the ambient space, not trying to triangulate the kind of that the data is somehow really living on. All right, so thanks.
Everyone gets it, I think. Jen gets it in writing. We have that. Thank you again so much, Peter. If you guys want to unmute yourselves and clap, please do so. And see you during the next meeting where Peter will take back over and be less incompetent.